Hey, it's Professor Dave. Let's talk about DNA transcription and translation.
He knows a lot about the science stuff. Professor Dave explains.
Now that we understand the structure of DNA, it's time to understand exactly how this molecule codes for a particular organism. How is it that a single cell containing a specific set of genetic material will result in the development of a fish, or a cat, or a human? To understand this phenomenon, we have to learn about transcription and translation. This is the collective process by which the genetic code is read by enzymes in order to produce all of the proteins in an organism. A chromosome is a very long molecule, consisting of many millions of base pairs. Most of these bases don't do too much, but certain portions of the chromosome are special. They are called genes. These are the parts that code for different things. In a human, a gene will be on average around 10 to 50,000 base pairs long, though the longest is 2.5 million base pairs. And when a gene is expressed, a specific protein is produced. So how does this work? The first step is called transcription. This is the process by which enzymes use one of the strands of DNA within a gene as a template to produce a messenger RNA, or mRNA. To do this, RNA polymerase, with the help of proteins called transcription factors, binds to a specific sequence within the gene, which is called the promoter, and pries the two strands apart. One of the strands will serve as the template strand, or antisense strand, meaning it will be used to generate the mRNA. And the other is the non-template strand, or the sense strand. RNA polymerase doesn't need a primer. It simply initiates mRNA synthesis at the start point and then moves downstream along the gene in a process called elongation, synthesizing the mRNA as it goes, reading the antisense strand from 3' to 5' and generating the mRNA from the 5' end, attaching RNA nucleotides to the 3' end as it goes. This is very similar to the way DNA polymerase synthesizes DNA as it moves along the template strand. The main difference here is that RNA is being synthesized, which, as we recall, will be ribose rather than deoxyribose, and it will have uracil instead of thymine. Unlike replication, RNA polymerase zips DNA back up as it goes, keeping only 10 to 20 bases exposed at a time. Once RNA polymerase reaches the end of the gene, termination occurs. The enzyme